Good morning, all of you. Today we will discuss single nucleotide polymorphism (SNP), a fundamental concept in genetics which has a significant relevance in pathology, diagnostics, and precision medicine. SNP represent the most common type of genetic variation in the human genome. Let's begin with definition. Single nucleotide polymorphism refers to single base substitution at a specific DNA locus, which is present in at least 1% of the population. So, two key points to remember. It involves one nucleotide change. It must have a minimum population frequency of more than 1%. If the frequency is less than 1%, we classify it as mutation and not as a polymorphism. Coming to molecular basis. The human genome consists of approximately 3 billion base pairs. Within its vast sequence, even a single nucleotide alteration can occur. Example, in a normal DNA sequence, if cytosine is replaced by thymine, it will lead to formation of SNPs. This may appear minor, but depending on its location, it can have no effect, subtle effect, or significant biological consequences. Coming to functional classification. This is the most important section from academic and exam perspective. SNPR are based on their SNP are classified based on their functional impact. First, coding region SNPs. When SNPs occurring in coding region, they can directly affect protein structure. For example, synonymous, silent SNPs. There is no change in amino acid sequences due to redundancy of genetic code. Second one is non-synonymous SNPs. These lead to the amino acid changes that are further divided into missense mutation and nonsense mutation. Missense mutation lead to one amino acid replaced by another, and nonsense mutation lead to premature stop codon, which causes truncated protein formation. Second one is regulatory region SNPs. When SNP occur in promoter or regulatory region, they do not change protein structure, but they alter gene expression level. They can influence how much protein is produced. Coming to clinical significance. Now let's understand why SNPs are clinically important. First, in disease susceptibility. Certain SNPs are associated with increased risk of diseases such as diabetes, cancer. They act as a genetic risk factor. In pharmacogenetics, this is a rapidly evolving field. SNP influences drug metabolism, drug efficacy, drug toxicity. To explain this, I will take an example that CYP450 gene polymorphism affect how drugs are processed in the body, and SNPs also have they also act as diagnostic and prognostic markers. SNPs are used in diagnostic marker in disease identification and prognostic markers in predicting disease outcome. Coming to application in pathology, from a pathology perspective, SNPs have multiple application. First, in molecular diagnostic, they help in detection of genetic variation. SNPs are helpful in risk stratification in identifying high risk patient, in targeted therapy, selecting specific treatment, and also in precision medicine where individualized patient care can happen because of SNPs. This represents a shift from generalized treatment to personalized medicine. SNP is a single nucleotide variation, but its impact ranges from a benign variation to major clinical sequences. Its importance lies in understanding disease mechanism, predicting drug response, and advancing precision medicine. Thank you.